I first want to introduce the project work for the very pragmatic reason that I know my style of lecturing and I tend to squeeze as much as I can in more than the available time, and that means I wouldn't have any time left. Okay, so the first thing is that we have a lecture room booked at 11 for the introduction of the project, which was a very good idea when I had the schedule made, except that I never checked to see where we have the room, and it's in a different building again. So we would have had to take 10 minutes or longer to walk there, then do the introduction, and then walk to the old building where we were yesterday to do the practice. That would take 1.5 hours out of our time, which is just a shame. So we'll not go to the Medical Faculty building at 11. We'll go to the practical rooms in the building you were at yesterday at 11. So just ignore that entry in the schedule. It was a good idea, but we've proceeded beyond this one. Okay. The other thing is that Bas has made the groups. Yay. And this at the same moment gives me the opportunity to walk you through a little bit briefly through the setup of Canvas. This is the starting page when you open the course. It will look a little bit different if you view it on your phone, because things are there's less space, obviously. But the information should all be there. So at the top you'll see the announcements. These are pointers to other stuff in the course. The most important one is, well, the schedule, but then modules, because all the stuff that you're going to do, the programming class, the practicals, biology class, lecture slides, previous exams for review. There's also a module for previous lectures, so if you want to look at next week's lectures, just look at the ones from previous year, and there might be some changes, but you get an idea of what's going to be there. Then below that you have a list of, it says course summary, but it's just a list of deadlines basically for different things. There were a couple yesterday, there is, pointed out, right? There's another one today for the tutorial 2 in the programming class, and so on. There should have been one for the reading for this today. I don't see. Oh yes, no, it's over there. It was yesterday at 11. Reading for lecture 2. I'll put in the reading ones for the other upcoming lectures as well. So I haven't had time for that. What we need right now is modules. And oh wait, yeah, otherwise it's going to be confusing. Student view. Otherwise you see. It's not that there's nothing in there yet that you're not supposed to see, but I just want to show you what you're seeing. Okay, so the intake is what you did yesterday. You can collapse this. More. Okay, go away. Yes. Lectures, old lectures. This is where we put our recorded lectures. We'll make a new playlist for this year. This is the reading. Right, okay. This is the project work. I just opened this one. If you checked the Canvas page earlier, you would not yet have seen this. Now you do. This is the one we want to look at now. I'll open it here, and then we can see. We have groups. We have 24 groups, lettered A to X, with 2 spare letters, so we can have 2 more groups in case we forgot people. If you haven't done the questionnaire yet, for whatever reason, don't worry, but it does mean that you're not assigned to a group yet. If that is true, find me or Bas, or ask any of the TAs to find me or Bas for you, so we can get you assigned to a group. So at 11, if you can't find your name in this list, come and find one of those. In addition to who goes with which group, there is a list of the other way around. So here it's easier to look up your name. You'll find you find the room you're going to be in, and you'll find your group. So Sam is going to be in F131 in group U, and he's going to work with the scope database. Not just him, but the whole group U is going to work with the scope database. Clear?
Yep.
Yes? So if okay, so if you're not on this list, you don't have a room to go to yet. In that case, go to what is convenient. Go to S329. Yeah, if you're not on the list, go to S329. Chalk.
Yeah, okay, but usually
Good. Yeah. Then the project. Where are my slides? The slides are over here. Cursor. Yes. Ah. Yes.
I cannot see the group. Yes, I don't have the pages.
No, you don't. You have the modules. So go to modules,
I'm at modules.
and then you should scroll down and you see project. Reload. Because as I said, I just opened it like during the coffee break. So if you already clicked on modules before, you wouldn't see it. Yeah. Okay. So we'll shift to the project introduction now, and then we'll shift back to evolution. So this is an evolving action. It has to survive. So that is the conditions. This is the list of people in the group, and then I thought, oh, it's actually much more convenient to put it on homology. So don't worry about it. Okay. So, who already knows what? Because we mentioned it before. Who remembers what you're going to do in the project? But I didn't say much about it yet. Anything? Nothing? So it's a seemingly simple problem. It's about homology detection. And to keep it even simpler, because there are lots of methods to do homology detection. Kim yesterday mentioned HMM models that are actually very sensitive for detecting remote homology. Because they use a lot of data from related sequences to make a profile by which you can recognize very distant routes. BLAST, Basic Local Alignment Search Tool, is the simplest homology detection tool that we have, and that's probably the reason that it's still the most well used. It is in the top 5 of molecular biology methods papers in terms of number of citations. And so compared to like ELISA, Northern Blots, those papers, right, methods, BLAST is in the top 5. So it's by far the most well-known bioinformatics tool. Also. Okay, SciBLAST is slightly improved version of BLAST. It's a little bit more sensitive for remote homologs. And the simple question is, which one is better? How do we do that? It's a question to you. How can we test, benchmark, whether BLAST is better than SciBLAST or the other way around? What do you in general need to, yeah.
Like optimizing the runtime?
Well, okay, you want the faster one is better. That's what you mean?
It's better in one way, but there's
Okay.
several ways.
So the first thing you need to do is specify what your optimality criterion is. And in this case, it is, I was thinking of homolog detection, right? How good is it at detecting remote homologs. Close homologs is easy, right? If two sequences are 80% similar in sequence, two proteins are 80% identical in sequence, then they're homologous. Yeah, there's no doubt about it. 30%, quite likely. 20%, possibly. 10%, maybe. What's the lower limit? That's the random chance of, if you have two completely random, unrelated sequences for amino acids, what is a random chance of random identity you expect between those two sequences? 5%. Why 5%? Yeah, right. So if you have an alanine in the first position on the one sequence, you have a 5% chance that by chance the other sequence also has an alanine there, right? Well, it's a little bit different because not all amino acids occur in the same proportion. Right? Alanines are actually quite more common. Tryptophans are a bit more are quite rare, and so they're different. Yeah, that's. I think in reality it's. Can never remember whether it goes up or down. I think it's a little bit lower in reality. Okay. So the question is, which of these two methods is better at detecting more remote homologs? More remote homologs being ones with less similarity at the sequence level. Yeah? Makes sense, right? I mean, in terms of evolution, the sequence similarity between two sequences is the amount of mutations that happened between them from coming back from their common ancestor. Right? So the less they are, the more different they are, the more distant they are in terms of homology. What is the definition of homology? Before we start talking about how to test it.
Sharing ancestry?
Yes. Yeah, so two sequences are either homologous or they are not homologous. They can be more or less similar, they can be more distant homologs or closer homologs, but they can't be a little bit homologous or a lot homologous. Yeah, they either have a common ancestor, which they both descend from, which could be very early, a recent event, or a very distant event in terms of evolutionary history, that either have a common ancestor or they don't. Right, so gene A and B, protein A and B, they're homologous because they have a common ancestor. Yeah? Okay. Yeah.
I thought the reading last night was suggesting that like all living things have a common ancestor.
Yes. That's a good, that's an interesting point. Yes. But we don't know. So so you could imagine that once upon a time there was a single cell. Maybe the cell is not the right word, but it was an enclosed environment where the first gene was copied from proto-DNA to two copies of proto-DNA. Two cells were made, and this was the first gene, and so on. It's probably more likely that there were a couple of these cells, maybe millions or billions, and they each started with more or less random initial sequence, or something like that. Right? Because if you try it once, it's very unlikely to work. If you try multiple times, it's very unlikely to only work once. Get my so if you have a mechanism that can generate something that starts life, and you try it only once, then you only get one common ancestor. But if it doesn't work, you don't have it. So if you have an environment that can generate the start of life, it can probably do it multiple times, and most of them will fail, but a lot of them might have been successful. So, okay. Very interesting question. Was not really in the topic for today. Yes, so if you go back far enough, deep enough into the depths of evolutionary time, these things get murky. Yeah. This definition works in most cases where it's used. Why are we interested in homology, by the way? That's what Kim also talked about yesterday. Why? We say we're interested in gene B, and we know it's homologous to gene A.
No function or structure of gene A is probably the same for B or
Yeah. So if you don't know anything about gene B except the sequence, if you don't know anything about gene B, you're lost. Right? If you know the sequence but not anything else, and you know, but you do know it's homologous or very likely to be homologous to gene A, then whatever you know about gene A is probably true for gene B. Its structure, its function, its cellular location, lots of things. But it could be wrong. Yeah, it's always a because things can diverge to the point where they have different functions, different structures, and so on. Yeah? Okay. How do we test this? What is the ground truth? How do we know for sure if BLAST tells us, oh, A and B are homologous? Or, no, that's not what BLAST tells us. What does BLAST tell us? Does it tell us whether two genes are homologous? What does it tell us?
Similarity.
Similarity between the sequences, right? So whether they're homologous or not, that's your choice. We set an e-value threshold and say, okay, well, anything above the e-value threshold, I'll be assuming it's homologous. So how can we test how valid that assumption is? And because if we can test that, we can compare whether SciBlast gets it better than BLAST. Or yeah, if you set an e-value threshold for SciBlast or for BLAST, whether everything above the e-value threshold or below, because it goes below, you need to be below the e-value threshold, better than the e-value threshold, is more often a real homologue for SciBlast than for BLAST. Then we know that SciBlast is better, right? How do we know whether something is a homologue?
We use a training set of known proteins
Known proteins. Is that good enough?
with known functions.
Known functions. Okay, so you could benchmark it on function. Now, if you know that these two have the same function, then it's more likely that you're right. No? Okay. Anything else you can look at? The ancestor. Right, do we have them? Usually not, right? In a few cases we have ancestor sequences. But, the thing about ancestors is that they are no longer here. There are previous generations, hundreds, thousands, millions, billions of years in the past. So if I say humans are descendant from chimpanzees, what do you say? Wrong. Why is that wrong? Humans and chimpanzees have a common ancestor that they're both descendant from. Yeah? So we have human sequences and chimp sequences, but we don't have the common ancestors. Right? That would be nice if we would have that, because that would be our ground truth. Right? If they're both similar to the common ancestor sequence, and you can sort of show this common ancestor is in the middle of the two. There's a couple of mutations that only happen here, a couple of mutations that only happen there. Yeah, then use that. So does your lack of response indicate that we can't do it? We don't know whether the proteins are homologous or not? Structure will help. Yes? Okay. So we have three things that we can look at, right? We have sequence. Unfortunately, we can do different things with sequences. This BLAST and SciBlast are just direct comparisons of one sequence with the other. Yeah? And SciBlast is a little bit more sophisticated. It uses a profile of related sequences. It's more or less the same thing. If you remember Kim's lecture from yesterday, you can review the video if you forgot. You can do it a little bit better with HMM models and inference of phylogeny. Right? Not just looking at the similarity here, but looking at the shape of the tree that connects these sequences in the way that we think is closest to how the evolutionary events have happened in the past. Wow, that was a complicated sentence. Did I get across? Yes? If not, review the video of today and ask me next week. Okay, so, right. We can look at structure. How does that help?
Usually the similar structure has a similar function. You will interact with the similar.
Okay, so similar structure has a similar function, right? Okay, so the third level is function. But why, if these two proteins have a similar structure, why does that tell us anything about the common ancestor? So why does a mutation not dramatically change the structure? Did everybody get that? Probably not. Okay. So structure, if they have a common ancestor, they'll probably have the same structure, because if you would have made a mutation in one branch that changed the structure, it's very, very unlikely that you would have kept the function. If you don't keep the function, you're likely to die. You're likely to be selected against. Yes? Okay. So structure is conserved better than sequence, because actually, I mean, evolution doesn't even care about function; it cares about survival. But some functions, some parts of function are crucial for survival. Some part of the structure are crucial for function, and some part of the sequence are crucial for structure. Right? So function is more conserved than structure is more conserved than sequence. Yes, so if we find similarity at the sequence level, it's very likely that these two proteins are homologous. If we find similarity at the structure level, it's even more likely that they are homologous. Yeah? And if we find similarity at the function level, then we have to be careful. Because if we only find it at the function level, and not at the structure or the sequence level, then how would we call that? Analogous, yes, and which is the product of not divergent evolution that you have here, but convergent evolution. Right? Evolution has to solve problems. Not true, but if it doesn't, you go extinct. Right? So the species that have survived, that we see around us, are the ones that have solved the problem, and sometimes you need to solve the problem again, because this previous solution was lost, or it was solved by a different branch of the tree of life. You don't know anything about that. So that's why octopuses and mammals have very different eyes. Both of them are good for viewing. How do we know that they are not homologous? They're not descended from the same common ancestor? Because octopuses have slightly smarter eyes. They have the light-sensitive layer in front and all the infrastructure at the back. We have it the other way around. So we have the light-sensitive layer, then we have all the neurons, then we have a layer of blood vessels and other stuff. If you would design a camera like that, nobody would sell it. It doesn't make sense. It evolved, it works. It's not a perfect solution, but it got us here, right? But the octopuses did it separately, and they did it. Same goes for proteins. If you suddenly find yourself out of glucose and there's only galactose, and you've just lost your galactose transcription factor that enables your galactose import mechanism, you either die or you find a solution, which is very likely going to be different than the solution you had before, but you lost. Yeah? Okay. So we can benchmark at the sequence level, and you have to think about why or how you can make this not circular. We're finding sequences by similarity. You can benchmark at the structure level, you can benchmark at the function level. Ideally, you use all three of them, but we don't have time for that in the project. So you guys are going to work with 3 different data sets: PFAM, at the sequence level, SCOP at the structure level, and GO for function. Yeah? PFAM stands for protein families. It's hidden marker models for protein families. You get a lecture next week or the week after about more about how these databases can be. SCOP is structural classification of proteins. So they're basically families of proteins that are very similar structures. And GO is the gene ontology. That's a structured database of vocabulary to annotate a gene function with. So there's actually two parts of Go. One part is the ontology, and the other part is the annotation database, where the terms are used to annotate gene functions with. Okay? Any questions about this? Is this clear? Yes?
Yep.
Okay, good. Now, a little bit about the planning. There's, hey, interesting. There's a step, here. Step one is setting up a compute environment. More or less already started dipping into that yesterday in the Linux introduction. You'll continue on that today. Then the next step is to find homologs using BLAST and SciBLAST. Then you have to create your gold standard database out of these databases, right? So like what you have to do with BLAST, BLAST doesn't tell you whether two sequences are homologous. BLAST tells you whether they have similarity in sequence, right? PFAM doesn't tell you whether two sequences are homologous. It tells you whether they are in the same family or not, or superfamily at different levels. Same goes for SCOP. Similar things go for Go. So depending, based on these databases, you have to decide on the information from the database whether you're going to call two proteins homologous or not. So you have a two-sided problem. You have to define homology based on your search algorithm, and you have to define homology based on your benchmark database or your gold standard database. If you're from computer science, this might be very confusing to you because ground truth is supposed to be ground truth. You should be able to rely on that. Well, welcome to biology. This is what we know. We don't have anything better than this. Well, there's other databases you can combine them, right? But if you can't get it out of the database, we don't know. Nobody knows, right? So it's the best we can do. So it's not a gold standard as in the perfect database. It's gold standard in terms of the best we have. It's the best we have. So that's the best we use. Okay. And then finally, when you have all of that, you can do the benchmarking and testing. And there will be a lecture later on about how to do rock plots and other benchmark evaluations methods. Okay. Then deliverables. This is for the for the bachelor students in principles. You'll basically go through all of these steps as well. You're only going to use one of the three databases. Laura, can you, or somebody can tell me which one? I forgot. Well, you're only using one of the three databases. And you'll go through this more as a practicals, right? So you have a little bit more instruction. You don't have to figure out everything by yourself. Okay. Then for fundamentals, for the master students, these are the deliverables. You have a draft report. You have a draft report. A week later, you have to deliver feedback on other people's draft reports. And then, based on feedback and your own evolving understanding of the matter, you'll make a final report. And then in week 7, if I'm not mistaken, we'll have discussion sessions where we mix groups so you can compare results and so on. We'll have 2 sessions, one about comparing BLAST and SciBLAST, and the other one is comparing benchmarks. Because there's a second research question popping up now. the first one we started off with was, is SciBLAST better than BLAST or not? The secondary research question is, which of these three benchmarks is a better benchmark to decide whether SciBLAST is better than BLAST or not? Yeah? Okay. So welcome to bioinformatics research. This is more or less the bread and butter of what we do. Yeah? You design a method, you find, test database, you test your method, and then you have to start worrying whether your test was good enough to show what you want to show. Yeah? Either you do it or somebody else will do it in their next mission. Okay. Any questions about this, about the project? We'll be around from 11 onwards and the other weeks as well, right? So you're on your own. So you can ask questions. Okay. Again, if you don't know yet which group you're going to be in, either because you're not on the list or you can't find yourself on the list for whatever reason, go to this group. Now, I have no questions about the project? Yes?
Basically, what I was telling is like if we get the data from the database and design a model to predict their homolog or not, and then we verify
No, you use BLAST and SciBLAST to find sequence similarity, which you use to predict homology. And then you take a criterion from one of the databases. So each group is assigned one of these two databases. So let's say you're in the scope group, so you're going to use structures, and then you need to set a cutoff in the hierarchy of the scope structure families, which level you consider homologous or not. You can be more than that. You don't have to solve all that now, yeah, but this sets you guys on the right track. Okay. More questions about the project? No? Find back my lecture. Yes, all right. Almost. Where are we? Yes. Yes. Okay. I have a few slides left and only a few minutes. Okay. Oh, we've done so much of this already. Good. Okay. If you see something whizzing, I'll sort of whiz through the rest of the slides. Most of it we've already covered here. If you see something where you're like, oh, wait a minute, that's important, and he's skipping over it, just raise your hand. Or yell. Okay. We've already talked about evolution. We talked about orthology, virology, and, well, I haven't talked about orthology and virology, but Kim did yesterday. If you're confused about it, look it up on the slide. Here the tree goes from the bottom to the top. Most trees actually grow that way. This is an interesting side effect, horizontal gene transfer, that's also very nice to know about. Mutations, yes, more mutations, types of mutations. Not all mutations were created equally, they were not created, they were not equal. Right, this is what I already covered, homology principle, yes, we already did that, right? Multi-searching, divergent evolution, reconstructing it. Good. Pairwise alignment. This is the way we compare sequences in bioinformatics. Who has not yet seen a sequence alignment, like the one that's on the wall? For who is this the first time they ever see a protein sequence alignment? Okay, a few people. And the DNA sequence alignment? So you can do this Kevin already mentioned this yesterday. You can align stuff at the DNA level or at the protein level. In some way, it should be equivalent if you're aligning the protein coding part of a gene or the amino acid sequence of the protein. But if the amino acid sequences are very different, the similarity is going to be very hard to catch at the DNA level, because you'll have different codons and you don't necessarily know the reading frame where to read. Yeah? So for distant relations, you look at protein sequences. But if you, on the other extreme, if you want to look at very tiny changes in evolution, you want to look at the DNA sequence, because some letters in the DNA can switch, and you still get the same amino acid. So you will be hiding those small, but they might tell you something about the microevolution of, let's say, an HIV virus in a patient. You can trace back how the population of viruses has evolved in a patient by looking at the DNA sequence. You can see a lot more detail there than you can see in the protein sequence. Okay. The game about alignment is to find out where to put the gaps. The gaps correspond to evolutionary events, like you lose an amino acid here, or you gain one over there, or the other way around. It's very hard to tell what, because you don't know whether the ancestor had a histidine at this position or not. If it didn't, this was an insertion. If it did, this is a deletion. Yeah? Because we often don't know, we call them indels. Doesn't solve the problem, just circumvents it. And the assumption here is that the very similar parts of the sequence are most likely to have been already there in the common ancestors. So you'd like to find these correspondences. Here. Good. There's correlation between the sequence and the protein, the structure and the protein. You'll find that most conserved parts of the sequence are in the inside of the protein. Why is that? Yeah, back. Because of the structure. So there's much more things you can break if you change an amino acid in the inside of a protein. Yeah? Think back to the car. You can change the color and the shape of the outside, but if you change something in the engine, it won't run. Yeah? Okay. And you can take out the seats. There's lots of stuff you can do on the sort of the outside of the car, and it's still a car, or at least it's still functional as a vehicle. Okay, good. Right, more structure. I'm going to go there. Transfer of information. We already did that. Function. Lots about function. Okay. This is maybe nice as an overview, and then I think I will stop there. So I introduced sequence structure function, right? So this is the main hierarchical organization of life. Yeah? We have sequences in the DNA, protein structures, RNA structures that somehow encode for the function. This is what we're most interested in. Why? Because it tells us why somebody gets a disease and somebody else doesn't, or why this drug works for this person and not for that person, or it works for this cancer type and not that cancer type, and so on. Right? That's all related to function. This is hard to measure. This is even harder to measure. Sequences are easy to measure nowadays. So what we really want is a way to go from sequence to structure, or better yet, from sequence to function. Yeah? And that's why we're doing all this. From a biophysical point of view, what almost all proteins can do, and we don't know how they do it, they go from sequence to structure to function. You transcribe a messenger RNA, it goes to the ribosome, you translate it to a protein amino acid sequence, it folds, it's functional. But there's some exceptions to this, and most proteins do it that way. If we would understand this process, we should be able to predict this, but we don't, so we can't. Yeah? So the best thing we can do is compare sequences of proteins and thereby transfer the functional annotation. If that fails and we have a structure, you can compare structures. If you have a new sequence, a new virus that break out somewhere, and you need to know what this new gene is doing in this virus, you first look for similar sequences. If you don't find them, you can look whether, if you can measure quickly the structure of this is a big if, of this new viral protein, you can compare it to known structures. Okay, good. With that, I think we have done enough for today, for the lecture at least. Are there any questions left for now?